Hello, this is Professor Torsen. This is the recorded lecture for Chapter 12. Chapter 12 looks at the origin of life on Earth. In terms of learning objectives, you should be able to define life and outline the conditions necessary to support life and the evidence that supports our theory on how life on Earth was formed. We are going to be looking at how to categorize species, so you should be able to understand the biological species concept and the morphological species concepts. You should also understand what a phylogenetic tree is and what it shows us. Know the differences between analogous and homologous traits. Know the differences between micro and macro evolution, and finally, how extinction can impact evolution. The origin of life on Earth can be divided into 3 phases. In the first phase, phase 1, we see the formation of small molecules that contain carbon and hydrogen. In other words, during phase 1, we see the formation of organic molecules. In phase 2, we see the formation of self-replicating, information-containing molecules. In other words, we see the formation of RNA. And then in phase 3, we see the development of membranes. Membranes are essential for life as they enable cells to compartmentalize things, which is necessary for maintaining various metabolic functions. So let's take a closer look at these phases, beginning with phase 1. Again, phase 1 is the formation of small molecules that contain carbon and hydrogen. Again, essentially molecules that are organic. The Urey-Miller experiment demonstrates the occurrences in phase 1. So the Urey-Miller experiment, which is shown in figure 12.2, took chemicals and molecules that were present in Earth's early atmosphere, things like hydrogen gas, methane, and ammonia. They put those chemicals in a flask and heated it, and some of those gases evaporated into this tube here. This second area on the right-hand side of figure 12.2 represents early Earth's atmosphere. So this chamber right here has those gases, hydrogen, methane, and ammonia. And they introduced electricity. And that electricity simulated lightning. This experiment shows that by introducing a little bit of electricity to these various gases, the formation of amino acids is possible. So what this experiment shows is that given the conditions present in early Earth's atmosphere, organic molecules can form spontaneously by the interaction of electricity and those gases that were present back then. Phase 2 saw the formation of self-replicating, information-containing molecules. In other words, the formation of RNA. RNA is a nucleic acid, and it is necessary to catalyze the reactions necessary for replicating nucleic acids. Some people view RNA as a precursor to cellular life. During phase 3, we saw the development of membranes. Again, membranes are necessary for cells to maintain their metabolic functions. And without the formation of membranes, we would not see the formation or development of cells. On slide 8, figure 1228 shows you a brief overview of the origin of life. Early life were small, simple cells that resemble prokaryotic cells like bacteria we have today. Over time, those cells developed into the organisms that we know are part of the eukaryotic family, which include plants and animals like us. So all life on Earth can be divided into 3 domains: bacteria, archaea, and eukarya. Again, bacteria are those prokaryotic cells, and eukarya or eukaryotic organisms have eukaryotic cells. These domains reflect the evolutionary relatedness of various species. So looking at this domain, I see that animal and plant cells are more closely related than, say, animal and bacteria cells. When we're talking about the origin of life and the development of species, we need to define what we mean by the word species. There are several ways to do this. We are looking at 2 different ways: the biological species concept and then the morphological species concept. According to the biological species concept, species are populations of organisms that can interbreed with each other or could potentially interbreed and cannot interbreed with other organisms outside their own group. This is called reproductive isolation. By using the issue of whether or not organisms can interbreed, we can define certain species. However, this limits our discussion to organisms that reproduce sexually. In terms of barriers to reproduction, there can be pre-zygotic barriers and post-zygotic barriers. Pre-zygotic barriers prevent the formation of a zygote, which is no fertilization between a sperm and an egg. Post-zygotic barriers are barriers that come up after fertilization occurs. So a sperm and an egg are able to meet and fertilize, and a zygote is able to form, but that zygote either does not survive very long or becomes an organism that is sterile. This slide 10 shows you examples of reproductive isolation. The pre-zygotic barrier, for example, would be individuals that are physically unable to meet with each other. That would be an example of a barrier that prevents the formation of a zygote. An example of post-zygotic barriers is shown on the right-hand side. In this case, a donkey and a horse are able to reproduce. They are able to form a zygote, and that zygote develops into a living organism called a mule. But this mule is sterile, and so there is no way to continue this species here because these animals are not able to reproduce. So again, this biological species concept that defines species according to their ability to sexually reproduce or not has limitations, because species that do not reproduce sexually, called asexual species, cannot be classified using this concept. This also causes difficulties in classifying fossil evidence because fossils don't provide enough information for us to determine whether or not two organisms could interbreed. Since the biological species concept has its limitations, let's look at what a species is in terms of the morphological species concept. The morphological species concept characterizes species based on physical features. Now, the choice of which features to use is subjective, but the advantage is that it can be used in species that reproduce asexually. In terms of when species separate and become two different species, that is called speciation. In allopatric speciation, a geographic barrier causes a group of individuals to be separated from the main population. So in figure 12.11 on slide 13, we see a population here that will be separated from one another through perhaps this river that's been introduced. Once those different populations are separated, these two separated populations can undergo changes in themselves that could lead to a point in time in which they are so different from each other they can no longer interbreed. Again, that is called allopatric speciation. The work that Charles Darwin did on the Galapagos Islands with respect to those birds, or the finches that he observed there, is an example of allopatric speciation. Darwin saw differences in these finches, especially in terms of their structure and size of their beaks. And again, that is due to the fact that these birds were separated on various islands that had different environmental features that put different selective pressures on them and caused the development of these different species from the initial species of finch. So phylogenetic trees, also called evolutionary trees, are a visual representation of how various species are related in terms of evolution. So again, we were looking at this bottom figure on slide 15 earlier, where we saw things within the eukaryotic domain, like plants and animals, are more closely related than organisms that exist in these different domains. So plants and animals are more similar to one another than, say, animals and bacteria would be. The closer together these organisms are on a phylogenetic tree, the more related they are. So these represent the common ancestors. So you can see rats and mice held a common ancestor here, but the common ancestor between humans and these small rodents occurred much further back in history. Evolutionary trees can be combined and constructed by using comparative anatomy as well as looking at DNA. So by using comparative anatomy, we can identify species as being similar due to their similar anatomy, again, in terms of their structures and how similar they are. The advent of various DNA technologies has enabled us to utilize DNA to determine the relatedness of various species. So here you can see there are 4 different species, and they are comparing the DNA of these 4 different species. Species 1 and 2 are more similar to one another and therefore suggest that species 1 and 2 are more closely related than they are to species 3 and 4. When we use comparative anatomy to determine whether two species are related or not, it is important to consider convergent evolution. So thus far, when we discussed comparative anatomy, we discussed homologous features. Homologous features, again, are features that have a common structure or a similar structure to one another and suggest a common evolutionary history. We saw this in the previous unit looking at the development of forelimbs in various mammals. So it looked at the forelimbs of humans, bats, I think dolphins were on there as well. And the idea that the structure of those forelimbs are very similar, the same types of bones with similar shapes. And again, these homologous features suggest a common evolutionary history. Analogous traits, though, are different. Analogous traits perform a similar function, but do not have similar structure or similar anatomy. So, for example, bats and insects both have wings that enable them to fly, but the structure of bat wings and the structure of insect wings are completely different and don't have any similarities to one another, and therefore don't suggest common evolutionary history. Evolution can be micro or macro. Microevolution is a change in the allele frequencies over one or a few generations. So small incremental changes is microevolution. Macroevolution, however, is the accumulated effect of microevolution over time. So it's looking at larger changes over a longer period of time. This is represented in figure 1222 on slide 18. When we consider how quickly evolution occurs, it is important to remember that the speed of evolution is not constant. It can be fast or slow. It can be continual or punctuated. And this is illustrated in figure 1223 on slide 19. We can see the top portion of the figure is representing gradual change in an organism. Slow and steady changes over a period of time to this particular species, as opposed to punctuated equilibrium, which is the bottom portion of this figure, in which we see an organism be unchanged for a relatively long period of time, and then a major shift, and then another period in which the organism does not undergo many changes. Adaptive radiation is a term that describes when a small number of species diversifies into a much larger number of species. Things that trigger these adaptive radiation are mass extinction events, colonization, and innovations in evolution. Extinction can be one of two kinds: background extinctions or mass extinctions. Background extinctions, which are represented in this green color on slide 21, shows you that over time there is a relatively low rate of extinction. Mass extinctions are when a large number of species become extinct all over the same relatively short amount of time. And these mass extinction events are usually due to a large and sudden change to the environment. So over Earth's history, there have been 5 major mass extinctions. And again, these are highlighted on figure 1225 on slide 21. This concludes the recorded lecture for chapter 12. Thanks for watching and take care.